Thank you very much, everyone, for attending today. Welcome to this seminar where we have Dr. Simon Martin. He's originally from South Africa, where he did his undergrad and master's studies, and then he later on moved to the UK at the University of Cambridge, where he did his PhD. And he was working on the evolutionary genetics of heliconius butterflies. So after his PhD, he remained in Cambridge for some more research, and he's currently a research fellow at the University of Edinburgh, where he's doing his research on Lepidoptera butterflies. So his topic is about that area of his in.
Great, thank you very much, Daniel. And I must start by saying thank you also to Beth Kaplan for kindly hosting me here, and Ian Gordon, who's my collaborator, who invited me out to Rwanda for my first time, and I'm really enjoying my time here and meeting people here at the university. And I'm really thrilled and honored to be able to give a seminar in your series. So, as Daniel said, I'm an evolutionary biologist, and evolutionary genetics is where I focus my work. And I'm interested in processes that alter the genetic makeup of populations and that either maintain or create new genetic diversity. And probably the most fundamental process underlying the origins of genetic diversity is speciation. So speciation is the origin of species. And if we think of the tree of life, you can think of speciation events as these branching points in the tree. This is a really nice example of birds called honey creepers that occur in the Hawaiian Islands that have speciated into really diverse and beautiful forms quite rapidly. So this process is really the most important processes if we're going to understand the origins of biodiversity on our planet. When I think about speciation, I don't think about it as an event, and certainly today we're not going to be talking about it as an event, but more as a process. And it's a process that can unfold over very long time periods. Is that it just involves change in entire population. Perhaps the simplest way to think about speciation is to think of two populations geographically separated, slowly evolve, either adapting to different environments or just evolving through random chance, through random genetic drift, to be distinct morphologically and ecologically. But the problem with this way of thinking about speciation is that populations often coexist as separate species. And so there's potential for interbreeding. And when we have interbreeding between species, which happens quite often in nature, we have a mixing of their genomes. So we can quickly look at what happens when hybridization happens. So here we have two chromosomes from two individuals, two red chromosomes from species 1 and two blue chromosomes from species 2. And we have a hybrid here. This is called the F1 hybrid, which receives one chromosome from each of its parents. And it'll obviously be a mix between these two parents. But what's really interesting about hybridization is actually what happens to these F1 hybrids. So as a reminder of high school biology, this is what happens during meiosis, the production of gametes. The two chromosomes in an individual first double, and then a process called crossing over happens, where two of them exchange material. And that means that we can get gametes that are recombinant. So recombination is the product of this crossing over process. And it means that some of our gametes will be a mixture of both parents' DNA. So in the context of hybridization, that means that when an F1 hybrid mates again, it's going to undergo recombination and say it mates with an individual from species 2. Now we have what's called a backcross hybrid, and this individual has a recombinant chromosome that's brought a bit of material from species 1 into species 2. So when this process happens, the boundaries between species can become blurred. So if we imagine two populations that come back into contact, they start mixing, their genomes become mixed, and over time, if there's nothing to stop this mixing, the two populations can become completely blurred and we'll have one population again, with no real genetic distinctness between any groups. So it's a very intuitive and kind of obvious idea that for speciation to happen, we need what are called reproductive isolating barriers or reproductive isolating mechanisms, something to prevent mixing between species. And when we study speciation, we're often trying to understand what the different types of isolating mechanisms are, what types of isolating mechanisms exist in nature and their relative importance. And this can be done by studying many different species pairs, but really every speciation event is a unique unfolding of a number of different evolutionary phenomena. And so I'm very fond of digging in deep to particular events, and that's what I'm going to be focusing on today. So just to first give you some examples of reproductive isolating mechanisms, perhaps the most obvious that might come to your mind is mate choice. So if the two species have evolved to a point where they no longer recognize the other species as a potential mate, then of course we are not going to get hybrids and we're not going to get recombination. The problem with this type, this isolating mechanism, is that if it breaks, so if an individual happens to hybridize, which does happen occasionally in many species, that individual, that hybrid, might have intermediate mate choice, and its choice could go either way. And so once you've broken down a barrier like this, it will break down further and further with more interspecies matings. And so it's not a barrier on its own. Another kind of barrier that you might have in your mind, which is a very strong type of organism, is hybrid sterility. So this appears in many species pairs, but not all of them. And one of the most classic examples is the mule, the hybrid between a horse and a donkey. And the mule is an F1 hybrid, so it is produced. It has one chromosome, one copy of each chromosome from a horse and one from the donkey, but it is sterile. It's unable to produce fertile gametes. And actually, if you look at the cells undergoing meiosis to produce eggs and sperm in mules, and compare those to horses, you'll see horses on the left here, we get perfect pairing of all the chromosomes. It's these nice thick bundles, whereas in the mule, the chromosomes don't pair up properly, and there's a complete failure of the production of the gametes. So this would be a very effective species barrier. But as I mentioned, this is not actually something we see in many species pairs, and it's not something that seems to evolve very early in the speciation process. There's a third kind of barrier that I'm especially interested in, called an ecological barrier. And here, there's no intrinsic problem with the hybrid, and there's no intrinsic process that prevents interbreeding between the parents, but instead there's an ecological selection acting against hybrids or individuals with mixed ancestry. So in this case, we have two species of Heliconius butterflies that I worked on during my PhD. On the left is Heliconius melpomene, and on the right is Heliconius sydno, and they have these bright color patterns that act as a warning to predators that they are toxic. So they have cyanide-related chemicals in their bodies, and so birds tend to avoid them. And predators, like this jacamar here, will have learned to avoid both of these color patterns. But the F1 hybrid, which is only produced in about 1 in 1,000 matings in the wild, is not something that most predators will have encountered. And so when this predator is evaluating potential food types, it would interpret the F1 hybrid as a potential source of food, as it hasn't learned to avoid it. And a colleague of mine, Richard Merrill, actually did an experiment with paper models of thousands of butterflies in Panama and showed that indeed this F1 hybrid is predated about twice as often of the parental species. So this is a mechanism that acts after the production of a hybrid that would prevent the mixing by selecting against these hybrids. But what's really interesting about ecological barriers is that they can act on specific genes. So in this case, the only genes that matter are the genes that affect the coloration of the hybrid. And so if we go back to our mixed populations, natural selection is only going to weed out those hybrids or those backcross hybrids that carry the incorrect combinations of genes for the environment they're in. So that's a subset of the hybrids. And in the long run, what this means is that the barrier between species doesn't apply to the whole genome anymore, to the whole chromosome, but instead to specific loci, specific genes on that chromosome. And so what we can get is what are called islands of speciation, where these two species or subspecies or populations are distinct at particular parts of their genome. Those are the parts where natural selection is effectively weeding out foreign genes from one species background. But in other parts of the genome, they're effectively the same population where their genomes continue to recombine and become genetically mixed. So to test these ideas, we obviously have to sequence whole genomes of wild organisms. And the organism that I'm going to be talking about today is the African queen butterfly, Danaus chrysippus, which is a very common species that anyone who lives here in Rwanda will probably recognize. It's a really beautiful and brightly colored butterfly. Its colors also act as warnings to predators, but that's not the topic I'm going to be focusing on too much today. What interests me about this species and what has drawn many people to study it is the fact that there is color pattern variation within the species. And this variation has some geographic signal. So as we move to the far south, west, north, and east corners of the African continent, we find very distinct looking morphs or subspecies. But these all seem to come together and interbreed in a large area here in East and Central Africa called the hybrid zone or the contact zone. And in this area, which includes Rwanda and parts of Kenya, Uganda, Tanzania, DRC, and so on, in this area, we can find all of these morphs, as well as hybrids between these morphs. So here we have a speciation, a potential species midway, which is always a great opportunity to study the barriers that form as speciation happens. So this is just an example of some of the diversity of color pattern in the species that can be found. These samples were collected not far from here in Eastern DRC, and just today we were looking at samples that were collected here on the campus in Huye, and we find all of this diversity here as well. So I was drawn to this species because there was a lot of pre-existing work already published when I started looking for a study system to move to, as I left from studying South American butterflies. And most of this work was done by two authors, David Smith and Ian Gordon, who's my host here today. And they've discovered all sorts of things about this species, but the thing that intrigued me the most is, in fact, here's just a snapshot of some of their papers. And what you notice when you start Googling about Danaus chrysippus is that the same two names, David Smith and Ian Gordon, show up over and over again. And so these are obviously the people to collaborate with if you want to make sense of this incredible butterfly. The thing that they have published that interested me the most is the fact that in this hybrid zone, we have a phenomenon called male killing, where male and female mate, and they produce a batch of eggs, but half of those eggs don't hatch. And the half that do hatch are females. And it turns out that the half that don't hatch are the eggs of the males that died before hatching. And there was many years of speculation as to why this might happen. And in the end, it was found that this is caused by a microbe, a molecule which is an endosymbiont, so it lives within the cells and tissues of the female, and it is maternally inherited, so it's passed from mother to daughter. It's also passed to the sons, but the sons who inherit it die. And so I'm sure you're coming to the same conclusion that I came to, which is that obviously these are populations that should have no males. And how does that work? And that is one of the puzzling questions about this species. But we do know that it's a very dispersive species. And so it could be that populations without males are replenished by male immigration from elsewhere. here we have a map showing the distribution of this Spiroplasma endosymbiont, the male killer. We don't have excellent sampling across the continent just yet, but hopefully we'll be able to fill this out in time. But the striking pattern that was already noticed a long time ago by David and Ian is that the male killing seems to be restricted to the contact zone, to this area where the species come together and hybridize, the subspecies. So there are a number of questions here about the causality. Did the male killer produce the contact zone, or did the contact zone produce the male killer? But the thing that interests me the most, and certainly interests all the collaborators that are working on this species at the moment, is what does male killing mean for speciation? So if all the males are dying in this contact zone, what does that mean for the movement of genes between these populations? So just a reminder, this is what we get when male hybrids mate. We get recombination, and we can produce all sorts of recombinant mixed individuals. But in butterflies, in fact in all Lepidoptera, there's an interesting pattern in that crossing over, this mixing of the chromosomes, doesn't happen in females. So a female hybrid that mates will only produce either pure individuals of one species or individuals that have totally intact chromosomes, but there's no mixing of the two chromosomes from the two species. Now, in our contact zone, the males are being killed by the male killer. And so, in theory, we have no longer any process by which the chromosomes of the two subspecies that are interbreeding, or the four subspecies that are interbreeding, can become mixed. And so this might be a new form of species isolating mechanism, not by killing all hybrids, but just by killing those hybrids in which recombination happens. And these females we can think of as a kind of a dead end in a sense, because they don't produce any sons, and their chromosomes never get mixed with anyone from outside their population. So is this driving speciation in Danaus chrysippus? So to answer this question, we wanted to look at genomes. And over the last 5 or so years, we've managed to sample the species from various parts of Africa and sequence their genomes. And the method that I'm going to be talking about today is a method for quantifying the strength of a barrier using genome sequence data. And it's a very simple idea where we calculate a statistic called FST, which is a measure of genetic diversity within populations versus between populations. And if we have completely mixed populations, as we have here on the left, then the amount of diversity within is the same as the amount of diversity between, and so FST is 0. But if we have completely separate populations that are not genetically mixing, we have much more diversity between than we have within, and so FST is large. What gets interesting is when we look at these ecological barriers, where the extent of shared variation varies across the genome, and we get parts of the genome where this two species are distinct, and so we get this pattern where FST shoots up at these islands of speciation, but is otherwise close to 0. And so this is the pattern that we wanted to look at in Danaus to understand the barriers between these subspecies. So we sequenced genomes from several individuals from South Africa, Nigeria, and the eastern coast of Kenya, just outside of this hybrid zone. And this is what it looks like when we plot this value, FST, across the chromosomes. So the species has 31 chromosomes. And I was really shocked, and we were all really shocked to find that, in fact, there seems to be almost 0 barrier across most of these chromosomes between these populations, despite being quite far apart and separated by this male-killing region. There seems to be nearly perfect mixing of genes on most of the chromosomes. Of course, there are a few standout chromosomes that are totally different, and these are these islands of speciation, these pockets of the genome that remain distinct between the populations despite ongoing mixing between them. And we think that these must be involved in local adaptation to different environments. Personally, I think that the color pattern might be selected by predators in different environments, but there are, of course, other possible explanations for why we would get selection for different genes in different environments, but it's definitely only focusing on certain parts of the genome. The one part that I'm going to talk a little more about is chromosome 15, which you can see is almost half a chromosome remains distinct between these subspecies. So this is much more than just one gene. In fact, there are about 200 genes in this stretch of the genome that remains distinct. And when we zoom in, and we can use a clustering algorithm to tell us which population each individual belongs to at the genetic level. Sorry, I'm jumping ahead of myself. So one thing we could tell quite early on by looking at the chromosomes of all of these individuals is that 2 of these peaks are associated with color pattern. So David and Ian had already described 3 loci that affect color pattern. One is called A, one's called B, and one's called C, and they affect different aspects of the color. These were described by breeding experiments, but now with the genome data, we can show that A occurs on chromosome 4, whereas B and C occur together on chromosome 15. And that's what really made me interested in focusing in on chromosome 15 a bit more. So when we look at individuals with this clustering algorithm, we find many individuals that are perfectly clustering into a single population across half of their chromosome. So these individuals seem to show no recombination across this half of the chromosome. But we also find individuals in the hybrid zone that carry 2 different copies of this region of the genome, one from one population and one from the other. So these look just like those F1 hybrids that we were talking about, except only over part of their chromosome. So clearly what's happening here is that these populations are mixing. Their genomes are crossing over and becoming mixed, but for some reason on half of chromosome 15, recombination, this crossing over process, seems to be suppressed. And there are various possible mechanisms that can explain this, and we're busy digging into that at the moment. And I'm not going to say much more about that today, but it's remarkable that such a large part of the genome, half a chromosome, would not be recombining. And so it seems that there are a number of mechanisms that are acting in combination to keep these subspecies distinct just on this one part of one chromosome. So how do we explain the complete lack of a species barrier throughout the rest of the genome? So perhaps this chromosome 15 is involved in adapting to local environments, but the rest of the genome is not. And there are currently, amongst our team of collaborators, 2 hypotheses. The one is that perhaps there's gene flow between these groups that is circumventing and getting around this male killing zone. That's possible to test, but at the moment we don't currently know exactly how far individuals tend to disperse and what the main routes and direction of dispersal are. So there's still quite a lot of work there to understand just how much gene flow happens and how far individuals can move. The second hypothesis is that there is, in fact, a barrier caused by male killing in this region, but it's too recent to detect. So this pattern that we were looking at, FST, takes some time to establish. So if a barrier only established quite recently between the subspecies, it might not be possible to detect differences in the genetic variation between 2 populations. So to think about this, we have to think about this male killer and where it's come from. So this is just a cartoon example thinking about a bunch of females that existed in the population at some time in the past. And what I've put in color here are their mitochondria. So the mitochondria are passed from mother to daughter, just like the spiroplasma. And so we can track the inheritance of these molecules into subsequent generations. And we see that each mother passes its mitochondria on to its daughters. But the original female that became infected with spiroplasma, also passes on her spiroplasma to her 2 daughters. And as you can see, her mitochondria, the blue one, is perfectly co-inherited with spiroplasma. So there should be a perfect link between every infected mother and the and the mitochondria of all of her descendants. And so here we have a bunch of descendants that have all inherited the same blue mitochondria. So one way to understand where this spiroplasma has come from is actually to look at the mitochondria of these individual females in the contact zone. So this is a phylogeny made from the whole mitochondrial genome sequence of about 45 individuals, and they're sampled from different parts of Africa. And on right here at the bottom, we have red dots showing that these are the individuals that are infected. And what we can see is that they all have very, very similar mitochondrial genome sequences that all descend from a single great-grandmother female. And we can actually date the origin of that female to around 2,200 years ago. So it seems that this infection has only entered the population once in a single female, and her descendants have now spread to fill most of this hybrid zone with the same mitochondria and this male-killing microbe. So in some senses, this great infected grandmother is one of the most successful insects on Earth because of how many descendants she has left in this population. So is 2,200 years long enough to establish a strong species barrier? Well, just last night I started playing around with the expected value of FST over time. I found a nice equation for calculating this, and it turns out that FST does indeed increase quite slowly. In fact, 2,200 years appears here on this graph. So we really haven't had enough generations of butterfly breeding and genetic drift in these different populations to be able to detect a barrier between them. So it's entirely possible that the male killing is creating a strong barrier, but we'd have to sit around for another few tens of thousands of years before we're really able to detect it. So that's a bit of a shame, but it does explain this dramatic pattern that we have where there's just almost no mixing on most of the chromosomes. So for the rest of this talk, I'm going to turn my attention a bit more to this male killer and its rapid spread in the population. One of the things that David and Ian noticed is when you take an infected female, so here we have an infected female on the left, and you cross her to a male, and that male is usually uninfected, otherwise he would have died. And they have different color patterns. You see strange patterns in the inheritance of color pattern. So in this case, we have this East African form called Klugii here on the left, and yet none of the offspring inherited that allele. And from previous crosses, David had already shown that this form on the left should be dominant. And so what they concluded is happening here is that this allele on chromosome 15 that controls the forewing color, this black tip, is not being inherited by the daughters. They're only inheriting one of the two alleles present in the mother, and the other one has apparently gone extinct. But what they realized is that, of course, it hasn't gone extinct. It was probably inherited by those dead. And it's a specific half. It's a half that carries this dominant color pattern allele. So how is this possible? How is it possible that an autosome, a chromosome that should be equally likely to be inherited by daughters and sons, is only being inherited by one of the sexes? The dominant allele, whereas the daughters inherited the two recessive alleles. So it is being lost in those dead sons. But why is this happening? Why is one of the alleles being forced into the sons? And the current hypothesis for why this happens is something to do with the sex chromosomes. So in Lepidoptera, instead of the XY chromosome system that you'll be familiar with in humans, we have a ZW system in which females have a Z and a W chromosome, two different chromosomes, and males have two copies of the same chromosome called the Z. And that means there's a female-specific chromosome. So it's a bit like the Y, that's specific to male mammals. The W is specific to female Lepidoptera. And the hypothesis is that this W has become fused to one of the copies of our color pattern chromosome, chromosome 15. And specifically a copy that's carrying this recessive allele. And that means that when they produce daughters and sons, the daughters, by definition, will carry this recessive allele because the W creates the female, is what creates the females. And therefore, by definition, the sons are inheriting the dominant allele and being killed by the male killer. And so when we think about inheritance of the male killer in the population, we have to no longer think about just the male killer and the mitochondria, but also this fused W chromosome that must have also been present in this original infected female and have been inherited by all of her descendants. So this chromosome here is also super successful in this hybrid zone population. So one question that we've been scratching our heads about for some time is, what is the relationship between the male killer, Spiroplasma, and this fused chromosome that occurs in all females that carry the male killer? And at the moment we have two hypotheses, and we're still really looking for evidence to reject one and accept the other. So the first hypothesis is called the passenger hypothesis. And in this hypothesis, the fused chromosome, that's got stuck onto the W, is simply hitchhiking as the spiroplasma spreads through the population. Why the spiroplasma seems to be spreading through the population so fast is another question, and we have various hypotheses about that. But it could simply be that by killing males, the daughters of each brood get more resources for themselves. The second hypothesis is called the partner hypothesis, and in this model, the fused chromosome is actually somehow involved in this male killing process. And if there's a benefit to male killing, then the spiroplasma and the fused chromosome are acting together to increase each other's fitness and spread together, kind of holding hands like partners in crime through this hybrid zone. So we'd love to be able to rule out one of these two. And obviously the more exciting one of these is the partner hypothesis, that this fused chromosome is actually involved in the process. But to rule that out, we would have to transfer this infection to individuals that don't carry this fused chromosome and ask whether the male killing still happens. And that's an experiment that we are in the process of trying to get going. And hopefully we'll soon have some results telling us the answer to this either way. But if there's anybody thinking about a master's project and finds this to be a cool and interesting question, then do get in touch, because we're definitely looking for people with the ability to work with the butterflies to help out with this work. So one of the things that really intrigues me about this species and this whole situation is that this hybrid zone population of females now has a shift in the way color pattern is inherited. And I believe that this could lead to rapid shifts in color pattern from one generation to the next. And here's a cartoon that illustrates why I think this should happen. So here we have a population of infected females with the dominant allele here, giving them this orange wing, and of course the recessive allele fused to their W chromosome. And populations like this with infection can be almost 100% infected with almost zero males. So a population like that is really dependent on immigrant males for mating. And these immigrant males have to come from elsewhere, and they could come from somewhere with a different color pattern allele, such as Southern Africa. And if they do, then they're going to be bringing this blue allele into the population. And what happens is that all of their offspring are going to inherit that blue allele. And why that happens is because of the male killing. So this allele that's fused onto the W ends up being inherited by these new infected daughters of the next generation. But this other allele is lost in the dead sons and is replaced by the immigrant males. And what this means is that depending on where migrant butterflies have come from, the color pattern in these male killer zones could rapidly shift from one generation to the next. And I think this is a really lovely idea to test. There are certain populations, we've found one just south of Nairobi, where there's very, very high density of male killing. And populations like that would be the perfect place to track the color patterns over time, tracking where immigrants might be coming from, and then tracking whether the females of the next generation are getting rapidly shifted to have different color patterns. So this is another ongoing experiment that we'd love to do and get an answer. So that brings me to the conclusions of this story. So what have we figured out? So we now know that these subspecies are separated by islands of speciation rather than a genome-wide barrier. The male killing is too recent for the barrier to be detected. So there may well be a barrier, but we'd have to wait around for a few tens of thousands of years to know for sure. We are currently thinking about experiments to possibly detect whether there is a barrier more rapidly than just waiting for tens of thousands of years. The Spiroplasma and the fused W are co-inherited, and there was a single infected female that carried all of these things. And all of the infected individuals in the contact zone that we found so far are descendants from that one female. And they have spread rapidly. And the reason they've spread rapidly is less clear, but we certainly know that to reach these levels of frequency, up to 100% in the population in just around 2,000 years is actually, implies quite strong natural selection favoring these females, despite the fact that they have no sons. So what don't we know? Well, we don't know why this male killer, Spiroplasma, seems to be geographically restricted to the contact zone. One possibility is that it's still spreading, and if we tracked it for long enough, we'd start to see it showing up in new populations. So just yesterday, we did some PCRs in the biotechnology labs here in Huye, and we found that there is some infection in samples here in Rwanda, but it's not as common as what we see in Kenya. But it could be that the Spiroplasma is still spreading geographically. Another possibility is that there's something restricting it geographically, some ecological factor or some genetic factor of the butterfly population that isn't allowing Spiroplasma to spread more widely. And that's something we really would love to figure out. We also don't know whether the partner hypothesis or the passenger hypothesis is true, so whether this fused W chromosome is actually contributing to the male killing. And we don't know, as I showed in that last slide, whether these rapid shifts in color pattern are happening. But some of these questions on the right are answerable by experiments. And I'm really happy to have come here and made some connections, because I think that the connection with Rwanda, where we have clearly a very mixed population with migrant males coming from many different directions, is a perfect place to answer some of these questions as well. So with that, I'd just like to thank all of these people on the left are my co-authors, and I'd like to just reiterate thanks to Beth, and then several other people that have been instrumental in getting our work and my visit here working. And then my main funding comes from the Royal Society, but we've had lots of support from other institutions, including National Geographic Society and our partner, Impala Research Center in Kenya. And that's all for my talk, and I'm very happy to answer any questions.
Yes, thank you very much, Simon. Looks like we don't have questions in the chat, so I would like to invite people to unmute themselves. If you have a question or comment to this talk, you may feel free to ask.
Hello.
Hi.
Hi.
Good afternoon, Martin.
Afternoon.
Okay. Thank you for your nice presentation. I have a small question about genetics of butterfly. How long can you get the F1 if, for instance, you can do this in vitro? It means in the lab. how long it can take?
Oh, to actually set up these breeding experiments in the lab.
Yes. Yes.
Yeah. This species is actually really tractable for doing genetics because it breeds all through the year, and it takes just over a month, something like 5 weeks. I'm looking at Ian to get from an egg to an adult laying another egg. So it's quite it's not very different from other model organisms like fruit flies for doing these kinds of experiments. The bigger challenge is keeping a large number of individuals because they need some space to fly around. But we've successfully bred this male killing a lineage of male killing infected females for 7 generations previously. So it is certainly possible.
Okay, I have another one. Okay, well, here in Rwanda, especially at university, we do have a problem of teaching genetics. And sometimes you see this experiment, it can help students to understand where the F1, if you wanted to do F1 or F2, we got only those kind of exercises with Drosophile. When we were abroad, we had this kind of experiment with Drosophiles.
Yeah.
But can we have a little bit the possibility of having this kind of experiment for our student to get the patent of colors?
Yeah, I think that's an excellent idea. So I think it's a really well-suited system for teaching because they're very easy to find. We've been very impressed by the number of samples that the students here in Huye have been catching as part of the ongoing monitoring of the population. And so it would be quite easy to be able to catch wild females. And when you catch wild females, they've almost always already mated. So you can catch wild females and give them the host plant, and they'll lay eggs within a day. And then you can set up crosses. And one of the things I didn't talk about is you can tell often whether an individual is homozygous or heterozygous for these color pattern alleles. And so there's a perfect opportunity to teach about inheritance of alleles, but also about dominance and recessive alleles.
Okay.
So yeah, I think it would be great if that could be incorporated into the genetics program here.
Yes, thank you very much. And we can still hear from some more questions. Anyone, feel free to unmute yourself and ask.
I have another things again about you said that you will use the PCR to see if we have this kind of contamination. Okay, I can call contamination of
infection.
I don't know. How did you get how did you do this one?
You mean how do?
Part of the butterfly, you cut them and do extraction, whatever.
Yeah, that's a really good question. So where in the body of the butterfly is this parasite found, this endosymbiont? So when it was first discovered, it was discovered in the year 2000. And in that study, the author specifically looked at the ovaries of females, based on the idea that if this is being transmitted from mother to daughter, it should be present in the ovaries. But since then, we've been looking at genome sequence data from the thorax and the head, and we've been able to detect the presence of this parasite from those tissues as well. And I recently also, in one sample, was able to detect it in the leg of the butterfly. So we don't know if it's at the same concentration in all of these tissues, but it seems to be present throughout the tissues of the organism.
Okay, thank you.
Sure.
Hello.
Hi.
Yes.
Hello, Martin.
Hello.
Hello.
Can you hear me?
Yes. I have a question about the color pattern. I would like to know if the color pattern plays a role in mate choice, because we know for some species like Gambusia, they are attracted by the good male. By good male must have the orange. So, in this case of this butterfly, do they color pattern play roles in mate choice, or just female they are blind, or something like that?
That's a really excellent question, because of course that could also be contributing to the barrier between populations. So we do have some evidence, not from my work, but from sampling that Ian and David have done several decades ago, that there is non-random mating. So that individuals tend to mate with others that share the same color pattern as them. We need to do more experiments to verify just how frequent this is and how strong it is, and what exactly is the driver. It seems to be in particular related to the B allele, the brown allele, which is also on chromosome 15. That could affect mate choice. But it doesn't seem to be, as you suggested, it doesn't seem to be about mating with the fittest or best genes male, but rather it might be driven by mating with a male that has the local color pattern, because that might be a better way to guarantee that your offspring have the same color pattern as you. But I'm not yet. I think other people would dispute that and say that we don't know what selection pressures are acting on the color pattern in the environment. So it's not really clear why you would choose to mate with others with the same pattern as yourself, unless there's some cost to mating with those with a different pattern, and we're yet to know what that cost might be.
Okay. So another question. What about the predations?
Predation?
Yeah, because sometimes when the predations, they are when the species they are facing with higher predations, they change their color pattern in order to protect or to reduce the chance to be attacked by the predators.
Right.
So, is there the research talk about?
Just repeat the final part of your question.
I mean, those species with color pattern issues, sometimes when the predation is higher, they change their color pattern in order to be not be attacked by the predators. They reduce for example, sometimes they reduce the orange and then they increase the black.
So are you asking whether that has happened in this system? Yeah, so
I would like to know if in this species it occurs.
Yes, okay. So we're very confident that these colors, the orange, black, and white, are not a coincidence. That these are warning colors that we see in many different insect and also other things like snakes and frogs that are toxic. So it seems very, very likely. These, I didn't mention, these are milkweed butterflies. So they feed on the milkweed family of plants, and especially the genera Asclepias and Gomphocarpus. And this contains compounds called cardenolide glycosides, which are toxic compounds that would be very distasteful to predators. And so there's quite a well-established natural history observations showing that this species is avoided by most predators, and that the warning patterns would serve to warn and to educate predators that this is food to be avoided. Why there are different warning patterns in different regions is not clear. We see that in some other warning pattern species as well. It might simply be due to chance, but there may be other factors. So, for example, in southern Africa, they tend to get more brown, That's also a cooler part of the range. So it might be that the warning pattern is not the only selection pressure, but that there's also pressure to have wings that might be a bit more absorbent to sunlight in that part of the range. So we're not sure whether the warning itself is evolving or if they're just other factors.
Okay. So, because I just asked this question because sometimes some studies shown that there is highly chance to be variated in color pattern due to the climate change or environmental change, also in combination of the predations. And as we know, this color pattern, they are basis on genetics. So maybe I will read more about it and can have much more. Thank you.
Yeah, sure. I would encourage you to read more about it because I think in this species, the original interest in this species came from this big hybrid zone where we find all of the color patterns. And there was a lot of speculation as to why a toxic species would have multiple patterns in one region. Because it's most efficient to have a single pattern that the predators learn. And I think currently we assume that the main contributor to this is immigration, but there may be other factors as well that we have not yet considered. So it's definitely something to still think about and think about experiments to test these different ideas.
Okay. Yeah, maybe if you have long time here in Rwanda, maybe soon we can meet to Butale, because I would like to join the center and I have one project which is going to talk about to investigate on the moths. And I'm still thinking about, but we are still planning how it will go on. So it needs little bit genetics.
In which species?
We will start with the Libonibide, is nocturnal ones, those ones we can get at night. That is the ones we choose. And then we continue with the other ones later, because still the project will be working on the other species later. So we started with nocturnal ones. That one we caught at night.
I see. Yeah, I'd be happy to discuss that with you.
Okay.
Yeah, thank you.
In Rwanda?
Can you ask again?
If
okay, I will just
would like to keep time, Eric, if you excuse me, so that I will be happy to share Simon's contact with you, so that we can keep in touch. So we have one more question in the chat from Deo, and it says, this is a great talk. I have ecological question regarding the African queen butterfly. While observing African queen in the field, sometimes you may see only males. Does this have an implication on ecological side of things?
So that's a really great question. So my entire talk has been about why you might find only females in certain populations. But how could we have populations with only males? Because surely they would also go extinct. And you're right. So there are occasions where sampling gives either a male bias or an entirely male sample of many individuals. And I think Ian might disagree with me, but I would say we don't fully understand that. Certainly dispersal and migration is part of that story. So it could be that there are areas in which there are very few local individuals living, and when we find individuals, they're always immigrants. And we believe, and there's some recent data from Nyamata here in Rwanda showing that males are more dispersive than females. And so it could be that that male-biased migration into areas in which there aren't any existing females could create these occasional periods when we find only males in the wild. But we really need more sampling over longer periods in multiple regions to know that for sure. And the other thing we'd love to know is where did these males come from? And that's extremely difficult to know It's not like some species where you can mark individuals and capture them weeks and months later, because they can often fly very long distances. And so we're thinking about using isotopes in the wings that might differ from different regions to try to make some sense out of where the immigrant males have come from. And I think that's probably going to be the first clue to why we find these male biased populations sometimes.
All right. Thank you. I hope that explains everything. And it looks like we have our time up. So let's call it a day. And I would like to take this opportunity to thank again Dr. Simon for his interesting talk. And I would like to thank every participant into this talk. Wishing you a nice evening. Thank you all. Goodbye.
Goodbye. Thank you very much.
Thank you.